count and how it can be carried out practically. So first of all, we try to understand what is viable count. The meaning of viable, that is living. Now here what we are going to find out that whether bacteria are living or not, whether they are viable or not, whether they are able to form the colony on the next day on specific media plate or not. So that is why it is called viable. Here bacteria are in the living form. In which living form? They are giving colonies. So colony, if they are able to produce, if they are able to reproduce, if they are able to metabolize, means they are living. So in that way, this is the practical in which we require 2 days to carry out the counting of the bacteria. This is a method of direct counting of bacteria. Now here what we are doing, first of all we will be provided with a bacterial suspension, which will be exactly 5 ml in the quantity. Now after that we are going to make the dilutions out of it. Why we are going to make the dilutions out of it? Because the size of the media plate, that is nutrient agar media, is limited. And if we add the entire amount of the bacterial suspension, bacteria will not give you colony which can be countable, but it will be grown in the form of loan which can be of no use, and we are not able to count the bacteria. So for that reason, we are diluting the bacteria exactly, very specifically, one by one, and that is why it will give you the result in the form of 30 to 300 colonies. So this is the fundamental of viable count technique, that we are interested in the number of colonies which can be counted, which can be observed nicely on very small nutrient agar plate. Okay. Now first of all, what we are going to do, first of all we will carry out the first dilution. This is called first dilution, and in the first dilution what we are going to do, we are going to dilute out our bacterial sample. How? We have 4.5 ml distilled water blend, and in that we are going to add 0.5 ml of our bacterial suspension. So what will be the exact dilution? Okay, if we take 10 ml, okay, if we want to dilute 10 times the bacterial cell, then what should we do? We should take 1 ml of bacterial suspension. It should be added with 9 ml of distilled water. Okay. Now here what we have done, we have reduced the quantity to half of the amount. We are taking 0.5 ml of bacterial suspension, and it will be added in the 4.5 ml of distilled water. To reduce the labor, we are going to apply this particular method, and the dilution will be 10 times. That will be, we are able to write it here, 1 is to 10, and it will be is equal to 10 raised to minus 1. Okay, so we have diluted 10 times the bacteria, so we can write it 10 raised to minus 1. So our first dilution will be 10 raised to minus 1. Now what is to be done in the next step? We are going to plate it out. Now how? This is our first dilution, and we have to take here 0.1 ml of this first dilution, and we are going to add it in the top agar when it is already hot. In top agar tube, we are adding our first dilution with the help of pipette, that is 0.1 ml. We will shake this properly, and then after we will overlayer or pour this top agar in the base agar plate. We will allow it to solidify properly, and then after we will keep it at 37 degrees centigrade temperature for 24 hours. And on the next day, we are definitely going to get the very well defined colony which are separated one. So on the next day, our result will be like this. This is our agar plate, and here you can see very nicely the separated colony like this. And you are able to count this colony properly with your naked eye, and if you require some aid, then you can also go for colony counter machine. And with the help of this, you are able to count the number of the colonies which are there in your nutrient agar plate, and you will come to know that these many are the bacteria they have produced the colony on the next day. Now the thing is that, is it always true that one bacteria will give you one colony? No. Why? Because there is always different arrangement in the bacteria according to the different species. So why it is so? You know that Escherichia coli is found always singly. But in case of bacillus, what happens? They make the chains. What happens in case of cocci? They are also found in the bunches or in the chains. So if the one colony, single colony has grown, it does not mean that it has grown from one single bacteria. Okay? The possibility is that it may be a bunch of the cell, or it may be the chain of the cell, or it may be individual cell. So there are so many possibilities. So we can never say that one bacteria is equal to one colony. So what should we do? That instead of writing bacterial cell, we should write the another word, that is CFU, that is colony forming unit. Colony forming unit. It means that it is something which is given the colony. It may be one single cell, it may be two cells, or it may be so many. Okay. So this with this clarity, I want to tell you that our answer of viral count is always in the form of CFU per ml. Okay.